All right, let's dive right into this explainer and expose the one tiny calculation mistake that is quietly destroying the grade boundaries of top Cambridge students. Picture this. You open your exam paper, you see a question on hemoglobin, and you think, great, I know this one. You start scribbling down everything you memorized about oxygen binding. But when results they actually roll around, your marks just aren't there. Well, why does that happen? It's because there is a massive trap hiding right in plain sight, and today we're going to break down exactly how you can avoid it.
Now, you really need to pay attention here because topic 2.3, that's hemoglobin structure and quaternary structure with heme groups, accounts for a massive 15% of the total marks in your paper 2 exam. I mean, think about that for a second. 15%. That is literally the difference between passing and failing, or bumping that B up to an A. It is an incredibly high-yield topic, but surprisingly, it's also where examiners see the most avoidable point deductions year after year.
So, why do ordinary revision methods just totally fail for this 15%? The stakes are incredibly high. And here's the thing. Top-performing candidates don't just regurgitate facts. They demonstrate a connected, conceptual understanding that actually transfers to unfamiliar contexts. If you're just writing out flashcards and memorizing textbook definitions, you're kind of setting yourself up to fall right into the examiner's trap. The assessment framework for this topic demands logical, structured reasoning. Cambridge examiners aren't just testing what you know, they're testing how you apply what you know to completely new and honestly sometimes pretty complex biological scenarios.
Okay, let's move on to Section 1, the costly hemoglobin mistake. This is exactly where marks slip away.
Let's see how this builds. Pulling straight from the Cambridge Principal Examiner reports, here are the 3 absolutely most painful ways to lose easy marks. Number 1, forgetting to include units in your calculations. I know, it sounds so basic, right? But under intense exam pressure, it happens all the time. Number 2 is failing to double-check those units and the specific terminology you're using. But number 3, and this is the absolute classic mistake, is providing a generic answer instead of linking every single point to the specific scenario they gave you. If the question gives you a specific environmental context, like, say, high altitude, and you just write a generic summary of how hemoglobin carries oxygen, you are going to get 0 marks for those points. You absolutely must anchor your biology to their data.
Now, take a look at this definition. Understanding this is just your baseline. Read it carefully. The quaternary structure isn't just, multiple proteins stuck together. It's the precise arrangement of multiple folded protein subunits alongside their iron-containing heme groups. But look at that second half. It requires analytical depth rather than superficial breadth. To snag those top marks, you've got to apply a systematic framework to this definition. You have to explain exactly how that specific arrangement dictates its function in literally whatever scenario the exam paper throws at you.
I want you to keep watching because missing this framework means you'll completely miss the subtle clues examiners hide right in the stimulus materials. In just a moment, I'm going to show you the specific examiner clue that changes your answer entirely, a detail so incredibly small that most students just read right past it. But to actually spot that clue, we first have to upgrade the way you process these exam questions.
Moving to Section 2, the method marks strategy. Remember, process equals points. In Cambridge biology, especially for those complex analytical questions, you're actually awarded marks just for showing your analytical process. Yeah, you heard that right. Even if your final calculation or conclusion is slightly off, simply demonstrating a logical, step-by-step approach can totally save your grade. This 5-step procedural framework is your ultimate weapon for tackling complex data interpretation systematically. First, deconstruct the question. What exactly are they asking? Second, extract relevant data from the stimulus. Don't just glance at the graph, actually pull the specific numbers. Third, select your analytical framework, which in this case is your conceptual understanding of hemoglobin's quaternary structure. Fourth, execute your analysis with exceptionally clear notation. And finally, present your conclusion using the correct terminology. If you follow this standardized sequence every single time, you literally force yourself to be analytical rather than superficial.
All right, Section 3, flawed versus top band answers, because as we know, context is everything.
Let's apply our deconstruct, solve, evaluate method to a real Cambridge-style comparison. I mean, it's one thing to talk about frameworks, but it's another thing entirely to see it in action, right? Look at the answer on the left. Hemoglobin has a quaternary structure that carries oxygen. Sure, it's biologically true, but it is deeply flawed for an exam. It's entirely generic. It's just superficial breadth. Now look at the top band answer on the right. The 4 heme groups within the quaternary structure bind O2, creating the specific saturation curve shown in the data. See the difference? This student is questioning assumptions, identifying the exact components, those 4 heme groups, and linking the terminology directly back to the provided data. That is analytical depth. That is exactly how you secure the marks.
So, when you're faced with a table like this in practical work or a paper 2 data extraction scenario, your job isn't just to read the numbers. The numbers are literally just the starting point. Your job is to extract the relationships between oxygen partial pressure and hemoglobin saturation percentage. You need to check for reasonableness. Like, ask yourself, does this data fit the standard S-shaped dissociation curve, where binding one oxygen molecule literally changes the shape of the whole molecule to make it easier to bind the next one? If it doesn't fit that cooperative binding curve, well, why not? What are the limitations or alternative interpretations here? Top-scoring candidates use a table like this to make justified evidence-based conclusions rather than just mindlessly copying the numbers into their answer booklet.
Here's a quick hack for you. To make sure you never mix up your terminology under exam pressure when analyzing these tables, create your own mnemonic using the first letters of the key terms for hemoglobin's quaternary structure. Whether it's to remember the 4 polypeptide chains, the iron ions, or the heme groups themselves, having a simple mental hook will stop you from freezing up. When that clock is ticking down, a good mnemonic ensures you extract the right framework from your brain instantly so you can focus entirely on applying it to the context.
Which brings us to Section 4, the hidden exam danger. Let's look at what actually dictates the mark band.
We've covered the method mark strategy and exactly how to avoid the generic answer trap, but there is one final, hidden danger that can absolutely still cap your grade. Even with the perfect framework, misunderstanding specific command words will ruin your answer. If a question specifically says evaluate and you only describe, your biology might be absolutely flawless, but your exam technique will cost you the grade. To master this, you've got to check out our explainer on command words next. Until then, I want to leave you with this. When you sit down for your next test, are you actually answering the specific question the examiners are asking, or are you just writing down everything you know and hoping for the best?